All right, this is the second page. And up at the top, the question becomes, why is it important to copy every chromosome? Why do we take it into those sister chromatids and it becomes a doubled chromosome before cell division? And the reason is this ensures that each of the 2 new cells that are produced are going to get a complete, oh my goodness, you guys. A complete set of chromosomes. Because what happens is you have a parent cell, and after it's all big and grown and it's got double DNA, it's going to go through mitosis and divide into what we call 2 daughter cells. And of course, they're going to be smaller because they're basically half the size of the parent. So these are 2 daughter cells, and they're genetically identical. They're actual clones because they have the exact same DNA of the parent. Okay, and so then each of these daughters right here, this daughter and this daughter, they're going to grow, and then they're going to double their DNA, and they get ready to eventually divide into 2 daughter cells. And so 1 becomes 2 and 2 becomes 4. The cell cycle is something that just keeps repeating around and around and around. So it's the repeating sequence of growth and cell division. So anytime someone develops cancer, it's this cell division and this growth and everything in here that's gone bad. So there's checkpoints throughout this cycle where, we stop and say, okay, hold on, is everything how it should be? Should we move forward? So these are actually called protein regulators. And if that regulator gets a mutation in it, it could be a regulator that kind of stops cell division if there's something wrong. So if there's a mutation there, they don't know to stop cell division, and it just starts spinning around and around, and it gets all crazy. The thing to understand, and I actually make this kind of my own here. I'm going to draw this over here. This is mitosis up here. And mitosis is split into P, M, A, T. Prophase, metaphase, anaphase, telophase. Mitosis, remember, is division of the nucleus. After mitosis takes place and you've got the nucleus divided in half, now we have to split the rest of the cell in half. And that's going to happen through cytokinesis. And cytokinesis is division of the cytoplasm. Okay, so now we've got 2 daughter cells. That's what we said over there. 2 daughter cells are the results after cytokinesis. And now you're going to go through and we're going to go into G1, which is where the cell spends most of its life, S, and then G2. And the G's always stand for growth. Well, how do we make cells grow? They grow by adding cytoplasm, okay? More fluid to pump that cell up. So at this point, that cell looks like this, and it's got single copies of DNA in there. Once it moves into this S phase, this is called synthesis. And this is where we double the DNA, or we copy it. So after synthesis gets done, the cell would look like this, where it's got those chromosomes that have the sister or the double chromatids. Because now when we get all the way up to the top, once we get to here, we're again ready to divide and go into mitosis. So anything here in the green is what we call interphase. And it's kind of the in-between. An interphase is comprised of G1, S, and G2.